Good morning. I'm Janet Rossant, and I'm President and Scientific Director of the Gairdner Foundation. And I have the pleasure of moderating today's panel on COVID-19, the Canadian research response. Since 1959, the Gairdner Foundation has awarded its prestigious Canada Gairdner Prizes to some of the world's top biomedical scientists, whose fundamental science discoveries have changed our understanding and treatment of serious health problems worldwide. This is the second of the Gairdner Foundation's online Global Perspectives panel, following our successful panel in May, where we featured some of our laureates who are also involved in COVID research. But today we're going to explore some of the growing number of important Canadian COVID-19 research initiatives, including the urgent search for effective vaccines, better understanding of post-infection immunity, the social and economic impact on vulnerable populations, and how best to harness open science to share data and provide evidence-based policy interventions. I want to acknowledge the sponsorship of this panel by Genome Canada, a longtime friend and partner of the Gairdner Foundation. And we'll hear more from Rob Annan later on Genome Canada's Genomics of COVID network. We've got a number of speakers to hear from, and of course, this is really only a sampling of the many different COVID research programs that are going on in Canada today. So bear with us if we don't hear exactly what you hope to hear. We have 3 sessions in which each speaker is going to give a short talk, and then we'll have a Q&A addressed to those speakers. Those of you who are watching online, if you have questions that you want to address to any of the speakers, you can submit that via the chat function. We'll try to pick up some of those questions in the Q&A, depending on time limitations. This panel is also being presented in a French translation on the different channels. So, it's my delight. I'm delighted to be able to introduce Mona Nemer to start this session today. So Dr. Mona Nemer, as you all know, is the Chief Science Advisor to Canada's Prime Minister, the Minister of Science and Cabinet. Her mandate is to provide advice on issues related to science and government policies that support it. This includes advising on ways to ensure that science is considered in policy decisions and that government science is fully available to the public. And of course, she's been front and center in all the various coordinated science-based initiatives to address the pandemic. So welcome, Mona.
Merci, Janet. Thank you so much. It's a pleasure to be here with you. And Janet, thank you for your leadership of the Gairdner Foundation, and a phenomenal organization that recognizes health research excellence, but also contributes greatly to the science and research dialogue in Canada, as we can see from this panel. I had actually the pleasure, as you know, of sitting on one of the panels, the Gairdner Whitman Award panel, and I want to take advantage of this to congratulate this year laureate Guy Rouleau, who is on the panel and will speak later on. As you mentioned, my role as the Chief Science Advisor is an exciting one, but it's really it's beyond, I guess, a lifetime experience to be in this role in the midst of an unprecedented global pandemic like the one that we are experiencing with COVID-19. I want to start by saying how grateful I am to the many colleagues, several of them on this panel, who have been helping me throughout this crisis, who have accepted to give of their time and sit on the committees that are advising our government, on task forces that are leading the efforts, whether on the immunity task force, the vaccine front, and others. We're fortunate that our government is actually seeking science advice, and I have to say, using it as well. In fact, this pandemic has highlighted the role of science and research like never before. We've never heard so much about science in the public, among the public, and in the general media. I think it's now upon us to maintain this trust and this interest in science among the public by engaging in a continued dialogue. The federal government, as you'll hear from the many panelists, has so far invested over $2 billion for research and development on COVID-19, from ventilator production and innovation to vaccine and therapeutics, but also supporting basic disciplinary and multidisciplinary research. You mentioned the social dimensions, the economic dimension, as well as basic immunology and virology. In terms of how the policymakers are using the discoveries and the results of the research, it's like pretty amazing. It's almost like in real time. The discovery comes out about how the virus is transmitted, and boom, there is a recognition of it, and it changed accordingly in several government policies. So I think this too is very rewarding. I salute the researchers in Canada who have been at the forefront, actually, of the research and these discoveries, both in government labs. Within government labs, you'll hear from the NRC. Of course, the National Microbiology Lab of the Public Health Agency has been also front and center, and as well research in numerous health research institutes and hospitals and post-secondary institutions. Many of the panelists will talk about these research from genomics all the way to immunity and vaccines. But let me perhaps also remind us that many research activities have been severely disrupted during this COVID-19 crisis. So let's not forget that many of the science that is being used to inform us more about COVID-19 was actually originated in other fields, from chemistry, biology, immunology, all the way to engineering and medical devices. So as we go back to reopen the research labs and restart the activities, and hopefully that we will not face further waves, but in case we do face other waves, let's perhaps reflect on how best to manage our research enterprise and our researchers. For example, some have suggested that researchers and scientists ought to continue their work and be considered as essential services and extended maybe the same support as we have done for others, including the healthcare workers. I think this is a question that deserves some thinking and some discussion around it. I also think that we need to pause and think about the gaps in our infrastructure that this crisis has perhaps unveiled, but also on how best and more effectively we can coordinate research across our large country and across our disciplines and sectors. So I think it's going to be a rich conversation today. Science has helped us so far winning over the war against COVID-19, and I'm certain that the final battle will be won thanks to science. So again, thank you for having me, and I look forward to the conversation today.
Thank you very much, Mona. It's a great start and a lot of good messages for all of us. So I begin then with the first session, which is on Canadian vaccine efforts. And our first speaker is Dr. Lakshmi Krishnan, who is the Director General of the NRC's Human Health Therapeutics Research Centre. Dr. Krishnan joined the NRC in 1998, and as a research officer, she built expertise in immunology research and then moved on to areas of vaccine adjuvant development, host-pathogen interactions. And prior to her current appointment, she was the program lead for vaccines and immunotherapy, and then director of R&D for immunobiology. And NRC is actively engaged in national and international partnerships related to COVID vaccine development. So welcome, Lakshmi.
Thank you, Dr. Rossant. I would like to thank Dr. Rossant as well as the Gairdner Foundation for the opportunity today to present NRC's efforts in COVID-19 vaccine development. Next slide. For those of you who are not familiar with the NRC, we are 102 years old. We are the largest intramural research arm of the Government of Canada in various sectors. I represent the Human Health Therapeutics Research Centre, which is one of the 14 research centres of the NRC. Equally, we are a funder through the Industrial Research Assistantship Program to support innovation among Canadian SME. Next slide. From the beginning of the COVID-19 crisis, NRC has been instrumental in pivoting our expertise and activities towards supporting solutions for COVID-19. I will speak to two specific aspects on vaccines and biomanufacturing, but also to mention that we have a number of other programs that is supporting SMEs as well as community, both in the universities and other government departments. In fact, since March, NRC has responded to over 600 requests for technical advice and short-term support for our collaborators. Next slide. As the first speaker here on the vaccine session, I want to give you a very brief introduction of the vaccine landscape as it exists today for COVID-19. There is a plethora of COVID-19 vaccines that are being developed globally. On the left side of the panel here, you see a breakdown of the various technology platforms that are being put to use. And you will, through this presentation that you will hear about some of these platforms, and my colleagues will explain other platforms in the context of what is happening in Canada and what we are bringing to Canada. As per WHO, there are at least 16 different vaccine candidates that are currently in clinical trials. And it's interesting to notice the target product profile from WHO. Of course, safety will be primordial, but also it is clear that even a vaccine that's 50 to 70% efficacious will have a significant impact in the case of this crisis. However, most importantly, such a vaccine will need to be effective in adults, and preferably in the elderly, because these seem to be the target population that are severely affected by this virus. Finally, vaccine development needs to occur at pandemic speed. And we see this approach in the way vaccine trials are being coordinated in global cooperation, but also through adaptive designs to move swiftly from phase 1 to phase 3. Equally, vaccine manufacturing is being done in parallel, sometimes at risk, so that the capacity to produce population-level vaccines can be made available in time when such a candidate vaccine could be approved for emergency pandemic use. Next slide. In this context, NRC has been focusing on actually developing tools, reagents, as well as deploying its expertise and technology platforms to support vaccine pipeline through two of our programs for which we received extra governmental support, for which we are very grateful. Next slide. Firstly, we focused our efforts on producing large amounts of the spike protein. As many of you will appreciate, the spike antigen is the main antigen in most of the vaccine development efforts globally. It is a very difficult protein to produce, given that it's a very large protein, it's a trimer, and it has got quite a complex structure. At the NRC, we've been able to produce the spike protein in our own CHO platform technology cell line, and we are producing nearly 100-fold more quantities than what has been reported for other cell lines. And we are right now characterizing this into a reference antigen bank and have provided this antigen to many groups, both in Canada and globally, to support their R&D efforts. Internally, we are using this antigen to also develop single-domain antibodies because we have significant expertise, and we are collaborating with various groups, both for vaccine evaluation, but also to develop antibody ELISAs that are more quantitative and more precise. Next slide. Secondly, our level 3 facility at the NRC, in collaboration with the National Microbiology Labs, is acting as a secondary reference center for SARS-2 virus propagation. We have also established our own in-house PRNT assay, which we are deploying with our partners. We are in the process of finalizing a pseudovirus assay, both for SARS-2, but also for MERS and SARS-1. And like many other groups, the hamster model is working quite well in our hands, as well as we are focusing efforts to develop a mouse-adapted SARS-2 virus. Next slide. So with these tools and technologies in hand, we are able to help a number of different collaborators to advance vaccine candidates. And NRC is currently collaborating with 3 different companies in order to develop vaccines. And I'll talk more in detail about 2 of them. VBI Vaccines, which is a Massachusetts-based company that is producing a pan-coronavirus vaccine targeting not only SARS-CoV-2, but also SARS-1 and MERS, as well as CanSino Biologics, which is using a different platform based on Ad5 vector. My colleague Volker Gerdts will speak about the VIDO efforts, and NRC is collaborating with VIDO to help them with the scale-up production of their vaccine. Internally, we are also evaluating some of our proprietary adjuvants that we have developed, as well as we are developing some viral vector platforms. I would not speak to them, but if you're interested, please approach me. Next slide. The VBI vaccine candidate we are working with is an eVLP platform. What this is a platform that was developed by this company some years ago, where the internal core protein is used to create a structure, and then the antigens are presented in the lipid membrane or internally. We actually produce this virus-like particle vaccine using our own proprietary HEK293 cell line. And the company has previously deployed this technology for other vaccine candidates, and they have demonstrated that it is highly potent at inducing neutralizing antibody as well as evoking a strong T-cell mediated response. The advantage of the technology is that the antigens take on the native shape because you are using a virus-like particle structure, and you could also look at multivalency using this technology. Next slide. So it was quite logical for NRC and VBI to collaborate to develop a COVID-19 vaccine. And because coronaviruses are also enveloped, the spike protein is a trimer, the eVLPs encourage trimerization and native conformation, and thus are definitely suited for this type of approach. The potency of the eVLP vaccine has been demonstrated in phase 1 clinical trials for other vaccine candidates, such as cytomegalovirus vaccine. And the safety has been also demonstrated in over 400 safe exposures. And this is a long-standing collaboration between NRC and VBI, so we were very quickly able to pivot and retool to produce both the monovalent SARS-CoV-2 virus-like particle, but also the trivalent particle for both MERS and SARS. And in preliminary immunogenicity studies, this data looks very promising for induction of a strong immune response. And we are hoping by the end of the year, we would be able to progress to phase 1 clinical trial. Next slide. The second candidate we are working with is actually an Ad5N-CoV vector, which is an adenovirus vector. It's a recombinant replication defective viral vector vaccine developed by CanSino Bio. CanSino Bio had done extensive preclinical work in a number of different models, including collaborating with VIDO for the ferret model and evaluation of their vaccine candidate in intranasal route. They actually commenced phase 1 trials in China in March, and it is actually the first data for any COVID-19 vaccine to be published in Lancet in May 22nd issue. And currently they're undergoing phase 2 clinical trials in China in about over 508 subjects, and the interim results were actually quite promising. And just as of this morning, China is actually approved this vaccine for military use in China. Next slide. So a quick snapshot of the data that came out of the phase 1 clinical trial, which was published in Lancet. On the top panel here is demonstrated that at 3 different doses of this vector vaccine that was tested, it did induce a dose-dependent antibody response as well as neutralizing antibody response. More importantly, the vaccine induced a strong T-cell response, including induction of polyfunctional cells, which is shown in the bottom panel here, where you're looking at cytokine production by CD4 T cells from all the 3 different doses. So based on this phase 1 clinical trial results, which showed that the low and moderate dose vaccine was well tolerated, Canada approved a CTA. Next slide. And we are currently gearing up to do this clinical trial evaluation. This will be led by Dr. Scott Halperin and Joanne Langley of the Canadian Centre of Vaccinology. And what we have done is design an adaptive phase 1/2 trial that will expand on the study that has been previously done. Firstly, to evaluate the vaccine in an elder cohort of 65 to 85 years old, but also to evaluate 2-dose immunization regimen for both a low and medium dose. It is expected that the phase 2 will be expanded to a 600-subject study, potentially multicentric, to evaluate the best dosage for different populations. Next slide. In order to support this clinical trial, as well as to be prepared to produce a vaccine for emergency pandemic use, NRC is also retooling to produce the vaccine at our facility in Montreal. The Royal Mount facility currently has a pilot scale at 200 and 500 liters, and extensive experience of the team to produce adenovirus vectored vaccines. And currently, we are hoping that our first production capacity of 500 liters can be brought to GMP compliance by fall 2020 to be able to produce clinical material for the phase 2, phase 3 trial. And subsequently, we will expand our capacity to be able to deliver 100,000 doses per month, as well as fill and finish for drug substance by spring 2021. Next slide. So to conclude, what NRC has been doing is working on developing preclinical evaluation of vaccines, biomanufacturing, as well as supporting clinical trial for various candidate vaccines. I would like to thank a number of collaborators who have mentioned throughout my slides who have been part of this journey, and we look forward to contributing to this effort. Thank you.
Thank you very much, Lakshmi. It's a really good start to introducing the vaccine initiatives that are going on. I'd like to now introduce our next speaker, who is Dr. Volker Gerdts. He is the Director and CEO of the Vaccine and Infectious Disease Organization, International Vaccine Center at the University of Saskatchewan. Dr. Gerdts' research interests are in the area of neonatal vaccines for both humans and animals, mucosal immunology, and vaccine delivery and formulation. Currently, Dr. Gerdts' team at VIDO-Intervac are working on vaccine candidates and animal disease models for COVID-19. So welcome, Dr. Gerdts.
Thank you very much for the introduction, and thanks for having me. My apologies, I'm driving in the car here, so pulled over, so I'm sorry that I have to do this from the car. So what I wanted to talk about is quickly our research that's going on at Vito-Intervac and really highlight the opportunities for collaborations. We're very fortunate to operate the largest containment lab in Canada, and it's so big that we can easily facilitate many, many trials in parallel. And again, this is an encouragement to people to contact us and reach out to us and work with us together, use our facility for the research that's going on. Next slide, please. So for those of you that are not familiar with Vito, just one slide on the background. We're a research organization at the University of Saskatchewan, 45 years old. Over the years have developed a number of vaccines, of which you can see 6 of them were world first. We spun off 4 companies. We're ISO certified. We hold the largest patent portfolio at the university and have over the years trained more than 500 graduate students, published more than 1,000 papers. And most importantly, we operate currently, as I mentioned before, one of the largest biocontainment infrastructures in the world. Next slide, please. So this is the Intervac facility, as you can see it here. And just the brief summary, the facility is so big. We have 18 animal rooms there. We can house in total well over 1,000 pigs in level 3 conditions. We house anything in this facility from bats to bison. And now with the COVID-19, we're working on hamsters, ferrets, and then, of course, mice as well. And it is one of Canada's 10 major science initiatives. So it's funded by the MSI program. And that is really helping us in operating this facility. Next slide, please. So for the COVID-19 research, we've been involved in this since the first week of January. Essentially, at the day of the WHO declaring a new disease, I phoned McGill, who at that time was the Director General of the National Microbiology Lab, and offered our help in working on this disease. We have started our vaccine work early in January, and as you can see, over the days or last few weeks have formed one of the largest COVID-19 teams we have had ever so far. You can see on the left side many of the lab researchers, and then on the right side, many of our animal care people. I want to point out Daryl Felserano. If you click on it, there's an circle coming up there. Daryl Felserano is the lead scientist on this project. He's a coronavirus expert, and has been instrumental in driving this project at the moment. Next slide, please. Since its initial outbreak, we were the first in Canada then to isolate the virus in Canada. This was in partnership with Sunnybrook and also the NML. We were then quickly established the hamster model for it. We also, the ferret model, and we also now have the hamster model established, and we're currently working on other models as well. As I mentioned, we started our vaccine candidate early January, made in-house the vaccine, and then in March started to immunize ferrets with it. And I'll show you some of the data later on, how well the vaccine works. We're members of the World Health Organization expert groups, and then most importantly, really down there, and as you have heard in the previous talk, we are assisting others. We're offering our services to others, and you heard some of the companies that we did the animal work for. We have now received well over 100 requests to test candidate antivirals, vaccines, and therapeutics. Next slide, please. So for the ferret and hamster model, as we all or may have heard, the ferrets overall, they show a very mild clinical disease. However, you clearly see viral infection in the upper respiratory tract. So there is viral replication, and I will show you the data. It is one of the major readouts that we're using to evaluate the vaccines. The hamster model in many ways is similar to that. The clinical disease is a little bit more severe. There is infection, also replication in the upper respiratory tract. Sorry for the background there. Next slide, please. So this is the ferret model, the first trial that we did. So there is a high dose and a low dose that was used. This is just to establish the model. And you can see we followed these animals for a period of 2 weeks to see what they show in terms of clinical disease and so on. Next slide, please. And there you can see we find the virus in the upper respiratory tract. So there's a nasal wash. You can see the higher dose. There's quite a bit of virus that we recover back from these animals for over a period of about 14 days post-infection. There's a little bit also in the old swab, but then there is very little virus in other mucosal sites, which is confirmed now by many, many observations around the world that this is really a respiratory, at least the viral infection happens at the upper respiratory tract. That's the site where you find most of the virus. However, the clinical consequences might be found also in other systemic organs. Next slide, please. When we then look at the nasal turbinates in detail, you can see over there on the left side, that's really where most of the virus is. And then when you look at the lungs, at the lung pathology at them, you can see that there is some gross lesions in the lungs of these animals. And next slide. And pathologically indication that we have a pneumonia happening there. So there's interstitial pneumonia, influx of inflammatory cells. We see both macrophages and neutrophils coming in. And we'll do more detailed histology at the moment to really determine what exactly what it looks like and what's going on. Next slide, please. Now, in terms of our own vaccine development, next slide. As you've heard from Lakshmi before, and like many others, we're also focusing on the spike glycoprotein. So that's shown in yellow there. As we heard, it's a trimer, and so the confirmation is critical that we have the trimer configuration well established. So our vaccine is a subunit vaccine that is formulated with adjuvants, and I will show you data really highlighting the importance of these adjuvants for the vaccine. We purposely chose the subunit technology because that's a proven technology, but it also gives us the advantage to eventually produce millions of doses per month, which is needed as we rapidly need to ramp up our vaccine capacity. But the key for a subunit vaccine is really to have an adjuvant that enhances the immune response. And for this particular disease, it's critical that we have an adjuvant that induces more of a Th1 response or cell-mediated immune response. And I'll show you some evidence of that in a second. Next slide, please. So you can see here the vaccine was produced in-house between January and February. We then, as soon as we had enough, went into animals, into the ferret model that I just showed you there. So these trials were completed now. We currently have 6 trials going on in which we are testing the vaccine candidates in various formulations, doses, and so on in both ferrets and hamsters. We also then already engaged one collaboration with NRC, as Lakshmi was indicating, regarding the manufacturing of the vaccine, and we're getting engineering batches currently done, and then GMP material by the late summer. We also have an international partner who's providing the antigen to us in partnership with us. That allows us then to use these engineering batches to continue our safety studies over the next 8 weeks. And so we have worked with Health Canada on laying out a plan for these animal trials. And we're hoping to start our human clinical trials. It's also going to be an adaptive combined phase 1, phase 2. Again, also with Scott Halperin in Dalhousie, starting in October of this year. And with that, we're really hoping that Health Canada then will allow us to, sometime next spring, allow us to release the vaccine under an emergency authorization to target a population. And it would be our goal that the vaccine would be available to the general public sometime in the summer of next year. Next slide, please. And so this is just showing you some preliminary data from one of the first trials that we did. And again, we tested here 2 different vaccines, 2 different adjuvants. One adjuvant was a commercial adjuvant that has been currently used in a number of commercial human vaccines. And the purpose of that was to use an adjuvant that has a quick regulatory pathway forward. And the other one is an adjuvant that we developed many years ago here at Vito-Intervac as part of a larger project funded by the Gates Foundation. And this adjuvant allows us to drive the response towards a Th1 response, and also is highly effective after single immunization. So what you can see here in these groups is we can see the antibody coming up, and these are actually neutralizing antibody titers. And so these animals were immunized twice. You can see at day 56 when they had received their immunization, and we're seeing good titers in both groups actually. And then after challenge, even your control group comes up as expected. So we are seeing with both vaccines high levels of neutralizing antibodies. But if you click on the next slide now, you can see when we now look at the replication in the upper respiratory tract that only one of the 2 vaccine works, the one on the left side. There you have, out of these 8 animals, 6 animals that are completely protected. It's almost below detectable levels of virus in the upper respiratory tract. Whereas on the other one, the one although it induced high levels of neutralizing antibodies, we hardly see any reduction in the viral replication. So what this really highlights, and I think this is what a lot of people are finding around the world right now, that the type of the immune response is critical for this coronavirus vaccine. That's why we're using an adjuvant that drives it towards the Th1. So many vaccines that are currently using not these type of adjuvants may fail, and when they go into these animal trials later on. Next slide, please. So here are our timelines and where we're hoping to be. As I pointed out, we're in the middle of the manufacturing right now. Engineering batches are being made, and the GMP production is underway and will be available by late summer. That will allow us then over the summer to complete safety and tox studies. And as I mentioned, we hope to be in phase 1 in October, continue into early next year, and then as quickly as possible proceed with either an emergency authorization and then also phase 3 trials later on next year. Next slide. That's it. So thank you very much. I'm happy to discuss any questions you might have later on in the panel. And again, thank you very much for the invitation.
Thank you, Volker. And now I want to move on to our next speaker, who is Dr. Gary Kobinger. He is the Canada Research Chair in Immunotherapy and Innovative Vaccine Platforms at the Université Laval. He's also a professor in the Department of Microbiology, Infectology and Immunology. And he's probably best known for his key role in the development of a successful vaccine for Ebola. So, Gary.
Just brief, this is an image of the virus, and I wanted to remind people that most vaccine approach are using the protein in yellow, coating the outside of the virus, the SARS-CoV that is causing COVID-19 as a target, which means that they are using that yellow protein, so the spike protein, to stimulate an immune response that wants to be protective. So when the virus infects the individual that has been vaccinated, this response can the surface of the virus and then protect the individual from the infection. Next slide, please. So this is just to give you an idea of all the landscape. A lot of candidate vaccine are out there, the majority of which are being advanced and developed by the private sector. Next slide, please. And on the left panel here, which was shown just before, are all the different kind of vaccine. The vaccines are divided into two big branches. The inactivated and attenuated usually use the entire virus particle. So the virus itself is grown and either killed or partially killed. And then the rest are called subunit vaccine, and they are usually using, as I said, this glycoprotein and the virus called spike. And this protein can be included into a DNA or RNA vaccine. It can be included in VLP. It can be used protein-based, or just the protein is being used with or without adjuvant. So you see this on the left panel. And on the right panel, you see the funding from or the support from those different vaccines. So you see the industry as the major driver of most vaccine, followed by let's say academic and the public sector, so government. Interestingly, and I wanted to point this out, the not-for-profit is present, but in a very small minority. Although of very interesting to me is the fact that a large pharma like Merck has partnered with a not-for-profit IIV to develop COVID vaccine. Next slide, please. And I think this is very important because it is telling you that the land vaccine development is changing for using new model that I do believe will fill a gap that still exists in vaccine development. So I wanted to put my own, let's say, selected preferred vaccine out there. For different reasons, as I said, the RNA and recombinant protein, Medicago VLP, Novio, DNA plus electroporation, and of course mine, the one from our team with a lot of bias. And it's not so much the vaccine, it's also the device, but the concept, because this vaccine is being advanced through a not-for-profit environment where ultimately the vaccine will be given for free to any country that wants to produce it, with the caveat that they wouldn't be able to sell it for a profit. And this is a model that we think is just in line with the Canadian healthcare system, which is universal access. It's not free by no means, but it's universal access and it's not for profit. One of the struggle of a healthcare system like in Canada, the UK, and many other countries is that it's using tools from for-profit entities, and that's fine. We're not against for-profit by no means. But it creates a gap where some vaccines, some therapeutics are not being developed because there is no economic incentive. This is where we think that the concept, especially of for-profit, not-for-profit side, is very helpful. So I'm showing you at the bottom, with the permission of Medicago, their VLP on the right of the two square, the left being the wild-type virus. So you see it's a virus-like particles with that spike at the surface. In the middle is the Novio, so it's a DNA molecule. Just to remind you that DNA does produce RNA. So the RNA vaccine is not much different from the fact that it's injecting an RNA that already made. And this is on the right-hand side. You can see on the slides. Then next slide, please. Then I'll show you a little bit the new injector that we have developed at Université Laval. Together with the engineer from the Department of Mechanical Engineering. And it's basically a tattooing machine that we have modified to inject with a positive pressure. The reason we did that is actually intradermal administration of vaccine is the most potent way to administer vaccine. This is from over 200 clinical trials that were analyzed. The minimum what you can do is reduce the dose. So in an event like now, it's very important because if you produce what you think, let's say 100 million doses, and you can use only 1/10 of it because of the device you're using to inject it, then it means that 100 million doses becomes 1 billion doses. And then you can reach a lot more, of course, a number of people, reduce the costs, and so on and so forth. So this is a device that is compatible to any vaccine that is in a liquid form. Was basically it injects 100 injection, same as tattooing, except that the tattooing machine does not inject; it just pokes holes, and then the ink will diffuse through the skin. This one is injecting with a low pressure, 100 times per second, micro bubble. You could see it as nano bubbles of the vaccine, which increase the vaccine response by a factor of about 10-fold to 12-fold. So it means we can reduce the dose of the vaccine and have the same effect. What we also is, next slide, please, is an increase that leads to the choice of giving, for example, with the device, 1 or 2 and have an immunity that is comparable to a 4 to 5 dose regimen for vaccines. So what you see in the left panel, the blue line is with the device, where you have a pressure injection. Green light is just a normal tattooing. Without the injection, you see some increase, but it's not statistically significant. Over the black line, that is an IM, so intramuscular injection, which is most commonly used for vaccination. Keep in mind that some vaccines are being given still in the intradermal space, like the rabies vaccine, for one. One of the important limitation is you give 0.1 ml. This device can administer half ml or 1 ml in less than 30 seconds. And this is the video we're going to see. On the right, it's just to show you on the prime boost with the device in red compared to no device in orange, following still a prime boost with the same vaccine. This was an HIV vaccine. Next slide, please. So on the COVID side, this device being used with a DNA vaccine that was generated, an improved version that we spent 2 years to develop. And you see this is in mice, 2 different line of mice, bulbs at the top and black 6 at the bottom. And the left is a T-cell response, and the right is with the dots is the antibody response. This is looking at total antibody IgG. As we believe, like for Ebola, that it's a good, first good readout, as opposed to trying to understand if the neutralizing antibody assay plays a role or not. But neutralizing antibodies are pending on this. But what you can see from a 2-dose vaccine is that there is a good B-cell response on the right. As a matter of fact, the signal is saturated with sera that were diluted 1 into 800. At day 56. And you see, to my big surprise, on the left, especially at the bottom left, that is a black 6 mice, which are a bit better for T-cell responses. Responses that are actually, I've not seen since the AD era of adenovirus 5. So these are extremely high level for a DNA vaccine. So we're very confident with that vaccine that we are going to get protection. The protection experiment is ongoing, so we still have to make sure of this. But again, like I have not seen those responses since working with AD5 15 years ago. So next slide, please. There's a little video. If you can click on it, it will show you the tattooing part of the administration of the vaccine, however it works. Okay, it's okay. No, it's fine. You would have just seen like it's the same thing. It lasts about 8 seconds to administer about half ml of the vaccine, and there's no visible damage. There's no color, of course. There's no ink in those vaccination, and it was in ferrets. But again, what I wanted to highlight here is the fact that this vaccine is being developed within a not-for-profit that is called Gerdts. You can go check it out, www.gerdts.org. And it's a new concept that we hope will provide new tools in the development of vaccine and therapeutics, and that we are right now using for advancing this vaccine that is receiving funds only through competitive processes from government, also donation, and from private and anybody. And I think it's going to be interesting to follow. Thank you so much for your attention. That's all I had.
Thank you. Now, if we can get all the speakers back on the screen. we are running tight on time. I knew we'd be running tight on time, but I think a few questions from the audience and I guess from me. One that comes up a couple of times from our viewers is the issue around, as we move to clinical trials, issue of who is enrolled in the clinical trials. Are we ensuring that this covers males, females, different ages, and of course different ethnic groups? What's the makeup of the clinical trial populations? I don't know who wants to address that one.
Yeah, it's Volker here. I can start. So that's all being considered. The regulators are recommending middle age group first, so 18 to 55 years, and then we'll have both males and females equally involved in it. And these are all volunteers. And you'd be surprised, for our vaccine, we have received requests for people to volunteer from all the way from Vancouver to all the way to Halifax.
Well, I guess the other part of it was whether those are reflect some of the more vulnerable populations that do seem to be more affected by the disease.
Yeah, so if I could comment on that, we have designed, Dr. Scott Halperin's group designed the trials for the phase 1, phase 2. In discussions with the regulators, the first we will go with healthy adults, 18 to 55, but in an adaptive design, looking at the day 28 safety results, we could then immediately go to an older cohort, which would be 65 to 85, which would include some of the vulnerable populations as well. As well, to mention that the National Advisory Committee on Immunization is working in the background to try and come with some recommendation of what priority groups one should consider during clinical trials in order to design the clinical trials that would be effective in the subsequent target populations. So we would be looking for those recommendations to come from NACI over the coming months.
So I had a technical question, which is you talked about testing the vaccine, obviously, in animals where you can infect them with the virus. Is there a need to do a challenge test to get the humans to get the vaccine to market?
Yeah, if I can go ahead, some vaccines that are currently in the clinic did not go through animal testing, which is quite interesting, as a matter of fact. But I think most candidates will be tested and evaluated in animals. It's seen still as a very, very important aspect, especially for a new candidate vaccine that have no data in humans, where it becomes essential. But there is a vaccine currently in humans that have not been evaluated, or were being done side by side. But the clinical trials was started. I can also add that depending on the vaccine, you can go with different population, always with approval from the regulators. But the DNA vaccine, we had elderly population, 65 and over in Canada, in Winnipeg, actually, with the universal influenza vaccine. So it's possible to do this, but now it's a bit different depending on which vaccine, and every vaccine approach and every strategy has its own evaluation through Health Canada or other regulators.
So Gary, you talked about a very interesting model where you're running it through a not-for-profit and making the vaccine available to any country that can produce it. That's, I think, a very exciting model of how one can combine the sort of the rapid approaches of pharma with the ability to get it out there to the countries where it matters. But we're seeing a lot of sort of nationalistic behavior in this whole vaccine world. So how do we ensure that in Canada, she says, let's look at Canada, how do we make sure that we get access to the vaccines? Does it mean we have to have our own vaccine? Or are there going to be more models like yours, Gary, where this really should be, after all, a global initiative to develop the vaccine, but also a global initiative to make sure it gets out to everybody?
Yeah, you're totally right. And as the major point here is how is it going to work for the ones that are crossing the line first? How is distribution, accessibility? I think it's going to be more complicated than anybody anticipates. And I think new strategy like getting those vaccine available, producing some of those vaccine production, manufacturing is not that hard. For example, there's manufacturing of vaccine in Africa. So accessing the basic component is often the big challenge. But I will agree with you that, for example, this is why one of the things I didn't mention, but this is why I picked also the Medicago vaccine, because they are the only vaccine manufacturer at scale in Canada. So I think it's an important candidate to consider for Canadian, because without them, there will be zero. At the same time, there's NRC that is, and of course it's not me to mention this, but they are, I think, working very hard to build up manufacturing in Canada. But right now, for what is available, it's not that simple. And I think we'll have to be innovative to solve all these, and not just count on others to bring their vaccine doses here. Mm-hmm. Thank you.
Okay, any other questions for each other at this point? Or any other comments you want to make? I think we've covered a fair amount of ground. Obviously, there are vaccine initiatives going on worldwide. There's also a lot get reported in the press, and I guess we just have to wait and see over the next few months. But even since we had our last panel, where we know that earlier was on the panel talking about where things were at that time, and it's amazing actually to see how much advance has been in basically just over a month. So we've seen a lot of vaccines coming forward. So it's pretty hopeful that there will be a vaccine, and then we have to deal with the manufacturing and the distribution. So thank you very much, all of you, and thanks very much. Now we're going to move on. So the next session is going to be around broader population-wide issues and impact on different populations. And our first speaker is Dr. Rob Annan. Rob is President and CEO of Genome Canada, and he drives there the overarching strategy of Genome Canada, which is to promote the value and potential of genomics in Canada to major stakeholders and raising the public profile of Genome Canada and genomics nationally and internationally. And like all of us, Genome Canada has also got itself involved very heavily in COVID research, and we'll hear about that from Rob. So Rob Annan.
Thanks a lot, Janet, and thanks for the invitation to speak here today. I really appreciate it. Just a big thanks first to Mona for her opening remarks, and then to my colleagues working on the various vaccine projects. I know how hard they're all working on that, and it's encouraging to hear the good work they're doing. So as Janet said, my name is Rob Annan. I'm President and CEO of Genome Canada. I'm going to, my remarks today, I'm not going to have slides. I'm just going to tell you a little bit about CanCaGen, which is a big national genomics initiative we're leading. First, just a couple of words about Genome Canada for those who may not be familiar with us. We're Canada's national genomics research organization. We're an independent, not-for-profit dedicated to doing applied research projects in genomics right across the country. So we work in health, certainly. About half of our work is in health, but we work across sectors. So we also have a lot of projects in agriculture, natural resources, energy, environment, and so on. So as a model, we work with 6 regional genome centers to support regionally relevant solutions sort of right across kind of advanced life sciences research and biotech. So as a national organization like that, I think we were well positioned to support a lot of the great work that burst into being very early in the COVID pandemic. So I'm going to tell you a little bit about that today. Really what became Canada's COVID-19 Genomics Network, or what we call CanCaGen. So CanCaGen is a national network that is using genomics tools to support the public health response to COVID-19, as well as to prepare for future waves and outbreaks. It really was and is a grassroots effort. So it's led by scientists from coast to coast. And those scientists are in government labs, academic research centers, hospitals, and public health labs. So we got involved early on as we started to take notice of various things popping up across the country, and came in to help try to coordinate across the system, provide some administrative support to those projects, and then to work with the federal government to secure funding to support the work. That culminated at the end of April with a federal announcement of $40 million for CanCaGen, which really supports 2 main projects. A viral sequencing project that aims to sequence up to 150,000 viral samples, and then a host sequencing project, which is designed to sequence up to 10,000 human samples. So we really have 2 related but reasonably distinct projects that then intersect at where it meets public health, where it meets intervention, and certainly where it meets data and so on. The network itself is comprised of a whole bunch of partners, certainly including national and provincial public health labs, hospitals, research institutes, the large-scale genome sequencing centers we have in Canada, as well as industry. So a real network approach. We're not just working in Canada, though. We are similar initiatives are happening around the world. We have formal associations with initiatives in the UK and the United States, but also are contributing to large public data repositories where international data is being shared to try to help drive broad public health responses. So I'm just going to take a few minutes now to talk a little bit about each of the two big projects, the virus project and the host project. So the viral sequencing project, it has a few different objectives, but really the primary one is to generate viral sequence data that allows us to use mutations that accumulate in the sequence to support track and trace efforts, and to support those efforts both locally, nationally, and internationally. So a couple of very quick words to orient us, right? So the SARS-CoV-2 virus is simple, right? It's a short stretch of RNA wrapped in protein and whatnot. And of course, it gets into us, gets into your cells, and then generates a whole whack of new copies that then burst forth into the world to infect other people. That copying process, like any other living process, is prone to errors, right? So not often, but often enough. So like in all living things, these random mutations occur while the DNA, or in this case RNA, is being reproduced, and sometimes those will have an effect on the viral fitness. But often they're silent. Either way, they're data that we can use as information. So as the virus spreads, the mutations accumulate, diversify. That pattern can then be used to reconstruct the history of any particular sample. So just briefly, by way of illustration, you can imagine a patient, from the initial outbreak in China, in Wuhan, China. That patient infecting two individuals, one goes to Europe and one goes to the United States. Those patients will now begin to accumulate mutations independently. And then they will infect people in their local regions. Eventually, when, let's say, a sample turns up in Canada, we can actually look at the string of mutations in that sample and ask ourselves, does this look more like the virus from Italy or more like the virus from the United States? This is a broad principle that allows us to kind of understand how those mutations can be used to basically backtrack the virus. So that simple idea has led to initiatives worldwide now to sequence lots of viral samples and then share them in public databases. So to date, there are more than 50,000 sequences available in public databases, and those samples are being used to create these really sophisticated phylogenetic trees. So like a family tree that shows how all of the various samples are related by mutation. So if you haven't gone and checked out a website called nextstrain.org, I recommend you do it. It's great. It allows you to really visualize all of this and get a feel for it. So how are we going to use this for public health? Well, in Canada, we can, this will help us with local outbreaks, right? We can ask ourselves, what the source of the outbreak? Is it related to existing outbreaks happening in the community or nearby? Does this look like a new entry, a new foreign entry? Different outbreaks, are they related? Are they distinct? It also allows us to track transmission trends nationally, regionally, internationally. So it's really genomic epidemiology. It gives us a lot of information, which is really cool. But it's not the only thing. It also allows us to get a couple of other really important projects going. One is to monitor the virus's evolution itself, and then to link that evolution with potential changes in clinical outcomes. So are we seeing changes in terms of disease severity or transmission? And could that be related, in fact, through metadata to changes in the virus's sequence? It also potentially opens the door to identifying challenges with PCR-based tests. So if we start to accumulate mutations in the regions where primers are being used to actually do the PCR-based testing, we might start generating false negatives just based on mutations. So never mind, you can use evolutionary analysis to look at areas that might be good drug targets or vaccine targets. So viral genomic sequencing has lots of potential benefits, lots going on. So the CanCoGen project on virus sequencing is being led by Dr. Terry Snutch out of UBC as the chair of a national implementation committee. There's national leadership from the National Microbiology Laboratory, as well as public health labs right across the country. That work is also being supported by the regional genome centers that work with us, as well as hospitals and clinics. So a real national effort. So work is underway. We're clearing, there's a reasonable backlog of samples that we're working our way through now, depositing that information in public databases, making it available to public health labs, and at the same time setting up sustainable systems for both the current outbreak, but also potential future outbreaks. So a couple of quick words now about the other project. This is the HOST project. It's really designed to help us understand why it is that COVID-19 exhibits such differences in terms of kind of variability of symptom presentation between individuals. So everything from like an asymptomatic carrier at one end to lethal disease at the other end. So this project is really designed to help us understand the role human genetics plays in that variable clinical response. There's strong evidence already that genetics play a role in disease susceptibility and risk. One study out of the UK showed that genetics contributes about 50% of the variability in terms of disease severity, especially around certain symptoms. So now this project and similar projects like it worldwide are starting to hone in on what some of these genetic features might be. And that's important because it also helps us understand better the progression of the disease and how it works. So these biomarkers, these genetic biomarkers that are being identified, help in terms of diagnostics potentially. Perhaps there's linkages between more severe response and certain biomarkers, and thereby identifying patients that would need maybe earlier intervention. But also, as I said, understanding how the actual disease works itself. So we all know that this is a mystifying disease. So if we can identify possible targets or pathways, some of which may have known interventions, we'll be that much further along when it comes to treating the illness. So so this project is designed both for the short term, but also creating a database that over the long term will have ongoing utility as a database of human DNA related to COVID for future outbreaks and even future pandemics. This project is being led by CGen, which is the consortium of 3 large-scale sequencing platforms in Toronto, Vancouver, and Montreal. And they are also cooperating with studies internationally to add whole genome sequence analysis from Canada to international initiatives. Right. I think now going forward, I just want to conclude by saying that what we're doing, I think, is important, obviously, and has a lot of value, but will be most valuable, I think, with integration with the other initiatives we're hearing about today, both on the vaccine side as well as what my fellow panelists are going to talk about. And it's important to deal with the COVID pandemic, but also for future outbreaks. So we're learning from this and preparing ourselves to do better. And finally, just one parting thought, which is that, this is one of the reasons that we invest in science in the long term, right? That the Canadian response, what I've seen and witnessed across the country has been impressive and unprecedented. Genome Canada is celebrating our 20th anniversary this year, and we've done a lot that I'm certainly proud of. But this rapid response really was 20 years in the making. The ability to turn around quickly is because we've got the scientists and the infrastructure to do that. So I just want to give kudos to the government for its quick action, but also for the long-term sustained support of science over the many years it takes to build up the capacity to deal with an outbreak like this. So I'll end there and look forward to questions and discussion.
Thanks for your putting it in context as well, and the importance of long-term investment in science that really does play off in the long run. So our next speaker is Dr. Catherine Hankins, and she is Professor of Public and Population Health at McGill University, and she's currently serving as co-chair of Canada's COVID-19 Immunity Task Force, using her experience as a community medicine specialist and her time as the UNAIDS Chief Scientific Advisor. Several of our speakers, I should say, are also on that same task force. So welcome, Dr. Hankins.
Thank you. And thank you to the Gairdner Foundation for inviting me to participate today. I've been asked to describe the work of the COVID-19 Immunity Task Force, and we are gathering new insights every day as this virus proves to be more complicated than perhaps we first imagined. I was very encouraged by what we've just heard about the work in Canada in vaccine development. And Rob's presentation actually made me start to think of broader issues like ethical and legal issues, thinking back to HIV and criminalization of non-disclosure. So there's other parts to this, societal parts to this science we need to think through. Next slide, please. When Prime Minister Trudeau announced the task force April 23rd, he indicated that we would be looking at key questions like how many people beyond those we've already tested have had COVID-19, whether you're immune once you've had it, and if so, how long that lasts. And these are the key questions that we're addressing. Next slide, please. Our goals are to catalyze, support, and harmonize the design and rapid implementation of population-based studies that will generate reliable first estimates of SARS-CoV-2 immunity overall and in priority populations across Canada. We will also support laboratory studies to establish the advantages and limitations of immunity testing and related technologies, and generate practical insights into the optimum ways of using and interpreting these tests. Next slide, please. I'm privileged to co-chair the Immunity Task Force with David Naylor, who is known to many of you, and the Task Force Executive Director is Timothy Evans. We have 3 ex officio members representing the Government of Canada, Teresa Tam, Mona Nemer, and Steve Lucas. Again, people that many of you know well, including Mona, who's already spoken today. Next slide, please. The leadership group is composed of 17 leading experts in the field of virology, immunology, public health, healthcare, and laboratory science drawn from across Canada. One of our ex officio members, Mona, has just spoken, as has Gary Kobinger, and after I speak, you will hear from Carrie Bourassa and from Vivek Goel. Next, please. The leadership group meetings are also attended by provincial and territorial representatives who have been appointed by their deputy ministers of health. 3 of the 13 already sit as full members of the leadership group, as indicated here with an asterisk. In addition, Kim Elmsley from the Public Health Agency of Canada and Mike Drebbett from the National Microbiology Laboratory also attend. We are supported by the Task Force Secretariat, composed of scientific core content leads for testing and diagnostics, epidemiology and biostatistics, data management and analysis policy, as well as support, admin, and communications staff. Some of these are acting positions with staffing procedures currently underway. Next slide, please. We have 12 governance principles that underpin our work, and I will touch on them in this talk because they are key to understanding how we work. We partner in all of our work with key stakeholders from across Canada to identify priority issues related to serological testing and its application. The ethos we are establishing is for rigorous gathering and rapid sharing of data to inform Canadians and advance the broad public interest, overriding considerations of personal and group advancement. We aim to mobilize the best science and study designs in this rapidly evolving field and establish fair and transparent processes that offer all interested partners across the country an opportunity to participate in the task force. We will work with partners to ensure protection of privacy in data gathering and safe handling of vital specimens. Next, please. We want to collaborate with partners and use existing data and sample gathering capacity wherever possible to enhance cost efficiencies and to provide a central coordination for the task force that facilitates rapid development of studies, their effective implementation, and rapid reporting of results to key audiences. We promote ethical and sound participatory practices that engage relevant stakeholders from study design through to dissemination and application of findings. We will adhere to best practices regarding scientific publications while ensuring rapid dissemination of reliable and relevant results in the public interest. We are liaising with relevant entities in other countries and with international agencies to better understand SARS-CoV-2 immunity. We aim to communicate the leadership, membership, activities, and results of the task force with openness and transparency, which is what I'm attempting to do today. Next slide, please. We have established an executive committee to approve all task force investments and to ensure that fast and transparent processes for the development, assessment, and implementation of investment notes and proposals are followed. We set out the procedures to govern potential conflicts of interest of leadership group members. As you might understand, our leadership group members are the top people from across Canada. We want to ensure ongoing review of adjudication principles and procedures for adjustments as needed. Now, this includes developing and applying principles governing sourcing of proposals and publicly available procedures to guide adjudication of proposals. We are critically reviewing the final budgets for value for money, and we see a phased implementation of investments with a view to ensuring efficient use of resources. Next slide, please. We have 3 working parties. The testing one, led by Mel Krishnan, is focused on assay validation, procurement, neutralizing antibody assessments, and analysis. Field studies, led by Ogilvy and Jim Kellner, on cross-functional to understand levels of in the puzzle, and longitudinal cohorts, which are priority populations at higher or rapid spread in communities, workplaces, and other settings. Immune science, led by Charo Koshik, is focused on correlates of protection, humoral cellular immunity, and duration of immune protection. Next slide, please. The 3 working parties clearly overlap, so a coordinated approach is needed with alignment and data share for them to synergize. So we see harmonization across the population surveys and longitudinal cohorts being needed, with decisions on what key participant sociodemographic and clinical data need to be collected, what biological measurements are of interest, which antibodies, titers, and functional responses are of importance. And this is needed both to inform immunology study design and by the same token is inspired by the needs of the planned immunity studies. Technical harmonization across labs is needed for assay validation, quality control, and proficiency. Next slide, please. You may be wondering what we've accomplished in the last 7 weeks. Well, we have defined strategic priorities for field studies and modus vivendi for operationalizing them, and we've determined our core data variables. Our Indigenous Advisory Circle has been active, advising the leadership group. Review is underway now of high-scoring proposals in the CIHR Phase 2 competition that fall within the task force remit. Our first survey is now underway. Canadian Blood Services and HEMA Quebec will provide a first look at seroprevalence in blood donors. And we are having productive discussions with provinces and territories. We are procuring assays for them in exchange for data sharing. We're actively exploring studying stored blood in biobanks, sources of ongoing blood collections, such as antenatal blood, and existing assets to draw on, such as established cohorts or expertise, such as Stats Canada. Next slide, please. Key developments so far. Health Canada has authorized sale of 5 lab-based serological tests, and procurement is underway now for initial studies. Protocols for assay cross-validation and for dried blood spots have been developed, with protocols for alternative specimens, such as saliva and cerebrospinal fluid, in development. Cross-validation of assays will start shortly in collaboration with provincial public health laboratories. We've provided briefings to chief medical officers of health, deputy ministers, first ministers, regular briefing notes to the federal cabinet, and we've launched SeroTracker. Next slide, please. So SeroTracker is a knowledge hub that tracks and synthesizes findings from SARS-CoV-2 serosurveillance efforts worldwide. The SeroTracker team is a global collaboration with 20 researchers, data scientists, and economists from 6 universities in the UK, Canada, and the United States. The academic affiliations include Toronto, Oxford, Calgary, Harvard, Waterloo, and McGill. Next slide, please. So SeroTracker has an interactive dashboard that allows you to filter results by geography and demographics and by population to visualize changes over time. Public health officials and policymakers can visualize surveillance and overlay it with diagnostic tests to put the serology results into context for a better understanding of SARS-CoV-2 spread. Next slide. This just shows you the global update from last night for 25 countries. On the right, you can see the filters. we are really keen to start to populate the Canada map with data. Next slide. Please go check out the Sero Tracker and all the information about the task force at the task force website. Here's the link. It's easy to remember. COVID-19 Immunity Task Force. Thank you very much for your attention.
Great, thank you very much indeed. Sero Tracker website looks like a very interesting place to explore. Our next speaker is Carrie Baratha, and Dr. Baratha is the Scientific Director of CIHR's Institute of Indigenous People's Health. Through that institute, she leads the advancement of a national health research agenda to improve and promote the health of First Nations, Inuit, and Métis people in Canada. The institute is now operated from the University of Saskatchewan. So, Carrie, welcome.
Miigwech, thank you. Appreciate the opportunity to be on this esteemed panel, and thank you very much for the Gairdner Foundation for inviting me. So I thought I would take a different approach. I thank you very much, Kate, for providing an update, and I have just a little bit of a slide to talk about the Indigenous Advisory Circle. So I wanted to talk about cultural safety and some of the resources that we've been able to develop, and of course I'll talk a small bit about CIHR, but I'm sure that's well covered as well. So of course I have a 50% appointment as a scientific director and a 50% appointment as a researcher. My home base, of course, is at the University of Saskatchewan. As I'm always taught to do by my kukum, who is a pipe carrier, I always introduce myself. So I am Anishinaabe Métis. I was born, raised in Treaty 4 territory with my grandfather in Regina. I grew up in the inner city of Regina. So just a little bit briefly about me. I've been honored to serve Indigenous communities across Canada for almost 20 years now, maybe even a little bit more. And so I just wanted to start out by talking about cultural safety and what that means and why it's important, particularly in an era of COVID-19. So in March, when COVID-19 really started to hit, it was really imperative that we ask communities, how can we help? What do you need? What's important? And it was about, at the time, really culturally safe resources, getting culturally safe information out. So what does cultural safety look like in COVID-19? Well, it means that, again, as we serve community and how we serve community means that we literally do that. Communities often sort of are in a deficit situation where we're trained as researchers to go in there and say, we have the solutions and what are your problems, rather than flipping that and saying, you already know what the issues are and how can we serve in a strengths-based way. So we started to develop resources from a strengths-based perspective in a very rapid manner. We engaged using OCAP principles. Ownership, control, access, and possession of data are vital, including specimens as well. So we also, of course, include the Tri-Council Policy Statement, which was developed through the Tri-Councils. If you want to flip the page, please. So this is what one of our first fact sheets looked like. It's very wordy and it's not very useful for communities, but we wanted to sort of collate what was the information out there at the time. We engaged partners immediately, Canadian Virtual Hospice, All Nations Hope AIDS Network, and Les Filles, the Métis Women's. And those were sort of our original partners, and they're still with us, but we have many, many more partners that have grown. And this was just sort of to get a baseline, and it actually served to help many researchers and clinicians to just sort of get a handle on what was the original information that was out there that we could just sort of pull together. if you want to flip the page, then the first fact sheets that we put out for communities, you can see are very different. And the uptake on this was immediate and spread literally like wildfire. And we called it the Protecting Our Home Fire Strategy. So on the left here, you can see this is in English, and on the right, it is in Nakota. And we have translated into many, many different languages, all of the Indigenous languages in Saskatchewan, and now we're branching out across Canada and beyond into the United States, into New Zealand, Portugal, just all over the place. And this is the kind of information that people wanted at their fingertips as quickly as possible. If you, I don't know if you can really see it very well, but you can just see that, this is, again, very strengths-based information, that people find very useful. If you want to flip the page here. So, again, communities started asking for more than just fact sheets. They wanted videos. And, in our communities, cultural safety is really about looking at systemic issues. It's not about culture. It's about, breaking down barriers. And for communities, in our healthcare system doesn't serve communities very well. Communities are very afraid of bias and racism and discrimination. And so creating culturally safe materials is absolutely vital, and particularly in the era of COVID-19. Communities remember when they had experienced in residential schools experimentation on them. They remember that research was not very kind to them. They remember H1N1 and SARS experiences. And, we are learning from them, of course, but it doesn't mean that the trauma is not still there. So they look to us to develop videos that were that's something that they could relate to. And we often use a lot of humor. We've been through an incredible amount of trauma, intergenerational trauma, and colonization is not something in the past. It is something that is still very much alive. We still have an Indian Act. And so we have to understand those intersections between historical trauma from the Indian Act and other policies, and the ongoing trauma from policies that are still in place. And while we are making progress, and I don't want to focus on negative all the time, but just to understand the context that's there. So we use humor, and so our team at Morningstar Lodge made a number of videos with a lot of humor, but really good information. The uptake was amazing. We're in the midst of partnering with Native Women's Association of Canada, as well as Dr. Margot Greenwood's Public Health Agency of Canada funded. I'm blanking on the National Collaborating Center for Indigenous Health, and so we're doing a number of webinars together, and again, a number of translations, and the home fires toolkits, the 19 ways to protect your home fires, and those were again, the uptake was great, and we had a lot of requests. Can you do something specifically for children? Can you do something specifically for our elders? And so again, these are all culturally safe. Specifically for Indigenous communities, First Nations, Métis communities, and Inuit as well. We also have submitted grants at the direction of community partners. This didn't happen right away. This was, a few months down the road, where the community started saying, well, what happens when there's a vaccine? Will we even be in the queue? And also, we're a little bit, afraid of the vaccine. I mean, is it going to be the same thing that has happened before? Is it safe? Tell us more about this. So a vaccine engagement strategy is vitally important. Contact tracing, and also emergency preparedness strategy. Communities are really understanding that there hasn't been appropriate emergency preparedness strategies, and that includes everything I've just said about vaccine engagement and community engagement, and knowing that they need to be leading this. They are the ones that have the solutions, and we are simply there to support them. We've also partnered with Canadian Virtual Hospice on culturally safe grief fact sheets. There are just layers and layers of grief, and the Canadian Virtual Hospice has been launching a strategy to understand why grief is not seen as mental health, as funded by mental health. And so, really, that was shocking to many of us. So grief, I think understanding for Indigenous communities, when we aren't able to gather and undertake our ceremonies, when somebody passes away, we can't have our funerals, and add that on to the historic trauma, is something I think that many people may not understand. So there's just layers and layers and layers of this that is going to happen, I think, for years to come. If you want to flip this slide. These are our processing grief fact sheets, and then what can we do? Because we don't want to sort of, focus on negativity all the time, although it's a reality. We want to try and figure out, well, how can we focus on some strengths and resilience and moving forward in a good way. And again, these were done in partnership, not only with Canadian Virtual Hospice, but the 30-member elder panel. It's a national community research advisory circle. And we have these advisory circles in every research project that we undertake with community, and they lead the way. They provide the advice. I know I'm running out of time, so if you want to flip the this is I know it's really difficult to read, but I certainly can share all of these. But this is a foraging fact sheet, and this was undertaken by our leading elder. And again, it was like, you can get out on the land, and there's if you don't, you don't have to be an expert in this, but this can guide you. And these are medicines for us. This is our medicine in many ways. Just being out on the land is medicine for us. If you want to flip the page. So I also wanted to share, what CIHR is doing. We've made incredible strides, particularly I think in the past 4 years, in terms of ensuring that Indigenous peoples are indeed leading the Indigenous health research agenda in Canada. And I want to say, Dr. Strong has been an incredible leader, and ensuring that we are increasing investments in Indigenous health research, and that includes investments in COVID-19 research. We have implemented an iterative peer review process because it is sometimes very difficult to find appropriate Indigenous peer reviewers. We have a new definition of Indigenous health research so that we are accountable to Indigenous communities, and we can measure and evaluate what actually is Indigenous health research so that we can meet our minimum 4.6%, and of course, hoping that we can go beyond that. And we want to ensure that we are accountable in terms of building research capacity and strengthening that. And also, it's not on this slide, but we are also reducing barriers to ensure that Indigenous peoples and communities can hold and administer research funds. And, that is absolutely key if we're going to be in alignment with the TRC and UNDRIP, and that Indigenous peoples are going to be self-determining. So that is a priority, and I could talk more and more about other barriers, but I'll run out of time. If you want to flip another slide, I think I'm almost done here. I did want to talk a little bit about the Indigenous Advisory Circle, and in the spirit of UNDRIP and in the spirit of TRC, while the Indigenous Advisory Circle does aim to ensure that Indigenous and non-Indigenous allies provide the appropriate expertise and knowledge, to the task force, we have certainly, you can see, I think we have now 28 members who are all working towards providing that advice. It is clear that Indigenous people need to lead a serosurveillance study. We have developed a proposal that we hope will be funded through the task force, and we certainly want to leverage other funding as well. And we see this as a top priority. It has to happen. We have never been involved in a national serosurveillance study, and certainly now is the time. So I just want to leave it there and say a chimigwich for the opportunity.
Thank you very much, Carrie. And briefly, we are running short on time and we have some hard stops coming up. So I'm afraid we're going to be cutting the discussion a little short on this. But Carrie, I did have one question for you. You emphasized how you're involved in the immunity task force, but I've got Rob on the discussion as well. So has there been a close relationship between your institute and Genome Canada as they develop their genome analysis in COVID patients?
Yeah, we actually did have that in our original proposal, but we were unable to get that piece funded, the task force, which is understandable. So we hope once our proposal is funded that we will be able to get it funded. We have been in talks with Genome Canada, and so we hope that we'll be able to leverage that funding once our proposal is funded. Hoping, crossing fingers, that it gets funded.
Okay. So another question that came up on the immunity task force, which is sort of related really, is to this issue of can you access the broad population, especially those people who don't go to hospitals and go to labs and have their blood taken. So how do you ensure that you really access the broad population?
So I think we have a really good. I guess the short answer is that we have really good engagement strategy, which is vitally important. We aim to engage many of our what's the word I'm looking for? Many of our networks. We have lots of networks, and one of them are the NERs, of course. But if you, and I didn't, of course, have the opportunity to run through all of those folks that are on there, who all have many, many networks. So we have networks of networks that we feel we will be able to really have a fully national Indigenous reach, I guess. Yes.
Okay, and I think I am going to have to cut it there. I'm sure that all of you, if audiences or anybody wishes to follow up, you're all willing to engage on the email or any other social media. So thank you very much indeed. Now we're going to move on to our last session, and we are running tight on time. So I'm not sure. I was going to try and swap the speakers here, and I'm not sure if that has happened or not. So who is the next speaker? Is it Dr. Rouleau? It's Rouleau? Okay, so we are going to go to Dr. Rouleau. And this is, Dr. Rouleau is the director of the Montreal Neurological Institute. He's chair of the Department of Neurology and Neurosurgery at McGill, and director of neuroscience at McGill University Health Centre, and most importantly, perhaps here, co-founder of the Tannenbaum Open Science Institute. And he is the 2020 Canada Gairdner Whiteman Award for his work on identifying the genetic architecture of neurological diseases, but also particularly for his leadership in the promotion of open science. So, Guy.
Thank you very much. I hope you can hear me. So perhaps I will try to catch up on some of the time. So I'm going to talk about open science in the COVID-19 era. Next slide. So I guess, first of all, open science means many different things to many different people. So what I'm going to be talking about of open science is really where scientists freely and in a timely manner share data, information, materials, tools, and research results. And the aim is to reduce barriers to collaboration and accelerate discovery. Currently, science is practiced in a mixed way. There are open science practices and there are also closed science practices. Next slide. What we've seen in this COVID-19 pandemic is really an unprecedented adoption of open science in the biomedical field. We heard many of the speakers talk about various open science tools and sharing methods that exist. We've heard about nonprofits, so we've heard a lot of open science being applied in COVID in Canada. In fact, open science is seen as the best strategy to solve the pandemic and is embraced by virtually everyone on the planet. Next slide. So just a few examples of COVID-19 practices. So data sharing, and these are just a sample of many that are going on all over the world, some in Canada and many all over the world. So data sharing, real-time tracking of pathogen evolution. We heard next strain, we heard about that earlier. Sharing of all the SARS-CoV-2 genome sequences in less than 6 days after sequencing. GISED, COVID-19 related protein structures, the Protein Data Bank, COVID-19 Cell Atlas, the CZI, the Chan Zuckerberg Initiative, the Human Cell Atlas, the Wellcome Sanger Institute, demographic health and testing data. We heard about that earlier as well. The COVID Tracking Project and others, centralized entry points, the European Data Portal, the COVID-19 Resources Canada, CanCOVID. And not only is data shared, but material is shared. There's proteins, for example, the Toronto Open Access COVID-19 Protein Manufacturing Center at the University of Toronto. Plasmids are shared by company Adgene, biobanking initiatives, the UK Biobank, which releases the data on confirmed COVID-19 cases, which enables a lot of the genetic studies, some of which were alluded to earlier, and the Quebec COVID-19 Biobank, which was set up in the province. Next slide. And this is just one example, and there are many, many examples, but I just really want you to look at the beginning of the slide. So one has December 31, 2019, the first reports of respiratory virus outbreak in Wuhan. And then you have basically 10 days later, the COVID sequence of the virus is published. 3 days later, using this open data, a company, Moderna, basically decides on how they're going to do their vaccine. And you can go down the whole list, but you can see that phase 2 clinical trial has started in May 29, and I've heard that they will be starting a phase 3 trial shortly. So an amazingly shortened timeline, and we heard these shortened timelines when we heard about the vaccines today, when we heard about all the various data sharing. So this is going on everywhere, and it's a great example of how open and rapid sharing accelerates discovery. Next slide. I think this has also led us to understand some of the limitations of some of the emerging open science practices. So there's a tsunami of information. We're currently, I think it's 600 preprints a day on COVID-19. And the preprints are not always accompanied with freely available data or information. Anyone can write anything and submit it. And so that is, of course, a danger, because good science requires peer review and replication. And if everything is seen before there's been peer review and before there's been replication by people who don't necessarily understand the limitations of science, it can be a problem. It has been a problem. Some partial solutions are being implemented again. It's a very rapidly moving field. We've got volunteers weeding out the obvious junk from large preprint places like BioRx. They're adding disclaimers saying that this information is preliminary. It's not information that has been confirmed. Nonetheless, there are issues that one can see. I've chosen the hydroxychloroquine story because I think virtually, most people have heard about hydroxychloroquine. The President of the United States has said some things about this and has also, I think he's taking it as well. I don't know if he's stopped or if he's still taking it. But there's one instance where after all of these various studies, there was a paper that was published in Lancet, and I have it here on the right, basically looking at almost 100,000 patients that really seem to show that hydroxychloroquine and chloroquine have no benefit and might have some risk. But surprisingly, the Lancet retracted this paper a few days later. And in fact, when you look at the details of why it was retracted because one of the partners refused to share openly the data, and it couldn't be validated. So the other authors asked to retract the paper. So this is an example of a failure of being open, leading to a failure of science. Next slide. So I just want to say that the COVID-19 has really brought into the limelight open science and has illustrated the power of open science. But in fact, open science is something that's practiced by many, many. And we at the Neuro, as you heard earlier, basically adopted open science. If you look on the right side, we're a hospital and a research institute. And so what we do is we have open biobanks, so patients with medical conditions. We have information and biomaterials. And if you go down to the right, then we make all this information available, the data available. Of course, this is all within an ethical framework, which allows the open sharing. We create data, we put data, make the data available. We do research on this. We have an early drug discovery program. And the aim, of course, is to accelerate going from the medical problem, which is on the top, all the way around to medical solutions, which would be treatments for disease. Next slide, please. So just some open, we heard a lot of this whole seminar is about the Canadian COVID-19, but here I just want to mention a few Canadian open science practitioners that are contributing to the goal of solving COVID-19. There's the Neuro's Early Drug Discovery Unit. It's developing models, most of using patients-derived cells, and this is being openly shared, including the protocols. The neuroethical framework that I mentioned earlier and the open MTAs, material transfer agreements, have enabled accelerated implementation of, for example, the Quebec COVID-19 Biobank, which passed ethics in a matter of almost days, because it was based on a previously accepted framework. And VIMI, which is an antiviral discovery initiative, which was launched pre-COVID, actually, by Alan Edwards and the Structural Genomics Consortium, and it's modeled on the DNDI, the Drugs for Neglected Disease Initiative, using the SGC's existing open science framework. So in a completely open drug development program. What we've learned is that it's critical to have open frameworks on which that can be shared and can be adapted to adapt open science initiatives to different situations. Next slide. So I just want to conclude. So COVID-19 is basically the first large-scale adoption of open science in the biomedical field. Major lessons: we need to implement mechanisms to ensure quality of data and publications. And here, I think AI is one of the major tools that's being looked at to develop various levels of quality checks. The importance of coordinating efforts and of existing frameworks, and existing academic, institutional, and government policies need to change in favor of open science. I could mention there's the open pledge, which is a COVID pledge, which is where institutions would agree to share freely intellectual property for a certain period of time to be able to accelerate discovery. Many institutions were unable to sign on to the open COVID-19 pledge because of institutional policies. Often the institutional policies are basically reflections of government policies. And I think my last point is that we're seeing an unprecedented pace of discovery for really urgently needed for this disease that's plaguing us. But in fact, open science on the same scale with global collaboration I think needs to be universal. There are many other diseases that affect us, whether they be heart disease, whether they be brain diseases or cancer. And really, our patients and we really need this to move forward as rapidly as possible, find solutions to all our medical problems. Thank you.
So our last panelist is Dr. Vivek Goel, and he is currently, for another day or two at any rate, the Vice President of Research and Innovation and Strategic Initiatives at the University of Toronto and a professor in the Institute of Health Policy Management and Evaluation at the Dalai Lama School of Public Health. And perhaps relevant to this discussion, he was previously the founding president and CEO of Public Health Ontario. Vivek.
Thank you, Janet. And I think the role of the last panel is to help catch up on time. But it's also been a great set of discussions, and many of the things I was going to talk about have already been touched upon. It's a privilege to be on this panel with Dr. Rouleau, and I congratulate him on the Gairdner Whiteman Award this year. The picture on my title slide is the Fitzgerald building at the University of Toronto. You can barely see it now because of all the buildings that have been put up around it, but this is how it looked at opening just about 100 years ago. And one of the really interesting things is this was the site of the School of Hygiene, as it was then known, and the Connaught Labs, which brought insulin to market, many vaccines. And even though they didn't call it open science, they operated on the principles that have just been described by Dr. Rouleau in terms of making their products accessible at an affordable price around the world. And we really do need to work on these initiatives and bring the kind of capacity that we had with the Connaught Labs back to Canada. So I'm going to speak briefly about the role of evidence in policy and the need for research coordination. It builds on Mona's opening comments and just conclude with some perspectives on the role of science. So if we could get the next slide. This is work that I did with Ross Upshur almost 20 years ago, when we were part of a network that was looking at the use of evidence in healthcare. And one of the things we realized is everyone had a different definition of what evidence actually is. And there's certainly in medical sciences, there's a very predominant view for the hierarchy of evidence from evidence-based medicine with randomized controlled trials at the top. Within this conceptual framework, that really sits inside of what we have as quadrant 3 here. And I'd encourage you to take a look at this paper. There are many other different types of evidence in law, in philosophy, in social sciences, across a range of disciplines. And when we think about evidence-based decision-making at all levels, we have to think about the broad definitions of evidence. The other thing, as I reflect on this, have gone back and looking at a paper I wrote 20 years ago, this actually speaks to what Carrie would talk about as Indigenous ways of knowing, taking a much broader perspective on the world than a very narrow reductionist approach, which is what most of our science is based on. Next slide. The other thing that is really important is how evidence is used in decision-making. We often think of a linear process by which evidence is introduced and decision makers make a decision. Evidence is always considered within a context of many other factors. And if you put little emphasis on context for decision-making, we get to evidence-based medicine, which is appropriate if you're looking to pick the right drug for a particular disease. When you get to evidence-based policy, it's very hard to remove context altogether. And at the other extreme, we have political decision-making, which some would argue is all about considering what the contextual factors are. The goal here is to try to get to balance. Next slide. So a graduate student of mine, Mark Dobro, again, did a study on how different types of scientific evidence were used in the development of the cancer screening guidelines in Ontario. And he developed this framework for all the different stages at which evidence are introduced. You see the solid lines and the dashed lines. The fact that evidence can come into decision-making in a variety of different ways and from formal and direct channels. So it's not a simple here's the evidence that a scientist produces, and a decision maker goes away with it. There's a much more complex environment in which evidence gets used in decision making. And next slide, as Janet mentioned, I was at Public Health Ontario during H1N1, and we actually used this framework and applied it to the decision making processes in Ontario and Canada during pandemic H1N1 through a series of case studies. This was led by Laura Zella, who was a postdoc of mine at the time, is now a Canada Research Chair at the University of Toronto. And the key findings were that, especially in a situation like pandemic, as we're in right now, there's going to be an incredible amount of uncertainty. There's going to be disagreement in the science. It is moving fast. As Dr. Rouleau noted, there are retractions. Things are coming back and forth at a very rapid pace. We have models. Models are a type of evidence, and they rely on assumptions, but they've been treated as factual. And then we have ideologies or perspectives. If you just think about the fact that people looking at the same evidence who have an occupational medicine lens versus an infectious disease lens versus a population health lens will interpret and recommend different actions for the employee, for the patient or healthcare worker, or for the population. It's not that they're disagreeing about the evidence. They have different requirements, different ideologies, different perspectives that they're applying to that evidence. The other key finding from our study was that our system is incredibly complex. As we can see, decisions are made at different levels of government, and decisions are made in different departments. There's the role of public sector organizations, private sector. There is no single entry point for science into decision making, particularly in the structures that we have in Canada. What is different, and it's certainly the creation of the Chief Science Advisor role and the development of departmental science advisors, has actually created a potential new framework for bringing evidence into decision making, at least within the federal government. Next slide. So the next point I'm going to turn to is the coordination of research. Mona noted that there's been over $2 billion of federal investment, and I would expect that if we counted up all the other sources, we might get to $2.5 billion or perhaps even $3 billion of investments in research. Don't try to read this slide. This is just a list of the competitions that we have up on our resources page. There's so many, this will give you a headache if you try to go through all of them. And the question we have to pose is, have we had too many research competitions? It's a lot of burden on our applicants, a lot of burden on reviewers, the research staff, and the granting bodies. And we have to ask the question, are we doing the right research? Are we missing things? I just went on, as Dr. Rouleau spoke, and looked at how many hydroxychloroquine trials are underway or pending in Canada on the Health Canada website. There are 8. Do we need any hydroxychloroquine trials? I think that is the question we should be asking. And then there's a question of, how do we support all the research? The competitions tend to fund the operating dollars, but we have a shortage of certain kinds of facilities, such as CL3s. So we need more discussions like today that bring together and look at what everyone is doing. And my final slide, please. And so some of these platforms have already been mentioned, as has the Immunity Task Force. Cancogen was mentioned, and I know there's more things coming, a vaccine task force and so on. But I also worry that we're now creating a new set of silos in research, because there doesn't seem to be an overarching coordination across all of these silos. We're going to be in this for the long haul. We're going to need a very robust response. Just want to close with one thought. As a public health physician, I've conducted a lot of applied research, and I see the importance during a situation such as a pandemic of the importance of targeted research, such as the Immunity Task Force is supporting. But as a vice president of research over the last few years, I've been a strong advocate for the role of fundamental science. And we can't forget the importance of fundamental science. We wouldn't have all the great researchers that we've heard about today if we didn't have investments on an ongoing basis in fundamental science. So I just hope that we don't lose sight of continuing to make those investments, even as we focus on applied work over the next few years to deal with this global pandemic. Thank you.
So the importance of open science, the importance of fundamental science, the importance of reaching out to communities and really building an open environment in which across Canada and around the world we can come together to solve not just this current pandemic, but to look at some of the big global issues that are going to continue to face us as we move in the years ahead. And so now I'm going to turn to Dr. Michael Strong to finish our presentations today. So Mike Strong, as everybody knows, I'm sure, is the president of CIHR. He was appointed in October 1, 2018. And prior to joining CIHR, he was the dean of the School of Medicine and Dentistry and a distinguished university professor at Western University. And during that time at Western, he served as chief of neurology and co-chair of the Department of Clinical Neurological Sciences at the London Health Sciences Centre and at Western. And Mike Strong is also a friend of the Gairdner Foundation, who's served on a lot of our committees. So it's a pleasure to welcome you, Mike.
Well, thank you very much, Janet. I appreciate that welcome, and thank you to everyone at the Gairdner Foundation for organizing this panel. Clearly, there's no denying that COVID-19 pandemic has been a terrible tragedy. It's resulted in hundreds of thousands of deaths worldwide and produced a generational disruption in our society. It's also really brought home the value of science within, but also the fundamental underpinnings of our research ecosystems and how at risk they can be. I think Vivek was just referring to a bit of that. It's an important area that we need to be watching very closely, thinking about as we go forward. But in the midst of all of that, in the midst of all of this turmoil, science and its intrinsic value to virtually all aspects of our lives has really become readily apparent and in the forefront of our communities. From the ability to make sound policy decisions to the amazing global commitment to eradicate COVID-19. The world is crying out for solutions, and they're turning to scientists for the answers to those. Not just as researchers, funders of research, and as part of the health research community. You know, Canada was one of the first to step up to the science challenge because of the pandemic, and including we were the first nation to actually have an open call for the best Canadian research ideas. We asked the community how best to invest the money, and the community responded immediately. This was the phase 1 rapid response programs. Whereas in other countries and other major funders, what we saw were that some governments were more prescriptive about the research and compelled the research community to apply accordingly to that. So we really did look at a very broad constituency, turn to the expertise of our academics, including our agency partners, Genome Canada, IDRC, provinces. And in doing so, we were able to mount, over this period of time, 9 different research programs focused on the COVID-19 issue. So everything from over $160 million, including the most recent phase, $291 million for student supports, so there are $450, which was announced last week in terms of how it's going to be allocated to research hospital institutes, universities for support. And then, as Mona alluded to in her earlier comments, a much broader base where the intramural program is also looked as being a critical component of this, particularly for the viral vaccines. Remarkable about this is the speed at which it has been done. We're doing things in time windows now that we would never have done before. So to create from the first glint of an eye to actual delivery of a program and have money out the door within 5 to 6 weeks is unheard of. And the speed with which that happens means that we have to be very, very cautious, very careful about what we're doing to try and avoid redundancy or duplication amongst a whole range of partners to do this. Not that duplication is a terrible thing all the time. As scientists, we like to see that data can be looked at in different ways or generated in different ways. So, but it is something we have to be careful of. We also, as Vivek has clearly delineated in his comments, we need to be very, very careful that we do not allow the entirety of the research ecosystem to come apart. Many of us have built our careers work in areas of research of orphan diseases or diseases that would not normally have a lot of funding coming forward. And yet, if all that funding disappears and all that's left is the COVID-19, then we will have done a disservice to the country as a whole. So it's very important that as we move forward, not only do we focus, and again, as Vivek has pointed out, on how we will move forward in the residual of the pandemic, but also all the other work we need to do. So with that, I do wish all of you well. I look forward to continue work with all of you very closely in the days to come. Thank you, Janet, and tremendous morning.
Thank you very much, Mike. And I just want to thank all our speakers today. We've covered a lot of ground, but there have been some common themes and obviously some crosstalk that I hope will continue after this session between the people we heard from, but also between those people and you, the viewers. So this presentation has been recorded, so it will be available on our YouTube channel soon if you want to look at more details. So you can check our website to find that out. You can also look at our Twitter, Facebook, and Instagram accounts for the information on this panel and also for any future events. If you have ideas of global perspectives panels that you think Gairdner should be leading, just let me know. Meantime, stay well, stay safe. Goodbye.